Do this diffusion and osmosis lab on video. All right, so if you've viewed the first part of the Chapter 5 lectures that covers diffusion and osmosis, then you'll be great for this lab exercise. Remember that diffusion is just the spreading out of particles from high concentration to low until reaching equilibrium. Remember also that the rate of diffusion can be affected by temperature. The warmer the temperature, the faster the rate of diffusion. It can also be affected by size of the particle. The smaller the particle, the faster it'll diffuse. The medium will also have an effect on diffusion. Air is faster than liquid, and semi-solids, like jello, would be the slowest thing you can diffuse through. And finally, the concentration gradient. The larger the concentration of substances, the faster the diffusion rate will be. The smaller the concentration of substances, the slower things will diffuse. Okay, so let's take a look at the setup for the first exercise in your lab. All right, so first I have to explain what it is that our lab techs did when creating these tubes. So let's look at tube number 1. Tube number 1 was filled with a jello-like substance. It's called agarose, but we can just call it jello. It's a semi-solid, and that's the point. And once the jello solidified, then our lab tech added a liquid dye, or a dye that's been dissolved in a liquid, on top of the jello, and then they capped it with a screw cap. That way it wouldn't leak out. They repeated the same sort of setup for a second tube, also filled with jello, but this time filled with a different color dye, and again, also capped so it wouldn't leak out. And finally, a third tube was created, also with a jello, and filled with a liquid dye, but this was a third different dye, and that tube was also capped. So there you have it. We have basically 1, 2, 3 types of tubes. The only difference so far is the type of dye that was put into them. All right, so bunches of tubes were made, and then those tubes were separated into 2 groups. One group ended up being placed in the refrigerator, and the other group was left at room temperature for a period of 100 hours. This will be important later. So what happened in those 100 hours? Well, the blue dye is aniline blue. And if you look in your manuals, aniline blue has a molecular weight of 737.74. If you look in the manual, you'll see that the other dye is potassium dichromate. I'm sorry, I know that you can't see that very well, but it's all in your manual. And this one has a molecular weight of 294.21. And finally, the third dye is Janus green. Janus green has a molecular weight of 511.09. Okay, so we should take notice that aniline blue is the heaviest, potassium dichromate is the lightest, and Janus green is somewhere in between those two in its weight. So over the course of 100 hours, what exactly happened with these dye particles that were sitting on top of the jello? Well, over time, they started to diffuse into the jello. And let's say aniline blue traveled this distance into the jello. Potassium dichromate would have traveled a farther distance. Why? Because potassium dichromate is much, much lighter than aniline blue. And Janus green would have traveled not quite as far as potassium dichromate, but definitely farther than aniline blue because it's somewhere in between the molecular weight of both of those other dyes. That should make sense, right? This is basically applying what we saw in the previous slide, that the smaller the particle, the faster it will diffuse. The heavier the particle, the slower it will diffuse. Okay, but now imagine this. Imagine that we've decided to compare let's say potassium dichromate that was at room temperature for 100 hours versus the potassium dichromate that was placed in 5 degrees Celsius. And what we might find is that the potassium dichromate that was placed at 5 degrees Celsius did not travel as far into the jello as its sister that was placed in room temperature over here. So this is the 5 degrees Celsius one. And so now we are looking at a second factor that affects diffusion. We're now looking at how the temperature can affect how fast a substance diffuses. So if we forget about Janus green and we forget about aniline blue, we will have noticed that the potassium dichromate at room temperature will have traveled farther than the potassium dichromate at 5 degrees Celsius, right? Okay, so how would we have known that? Well, you could look at it visually, but what we would have wanted you to do is we would have wanted you to record this distance in millimeters. So how far did aniline blue go? How far did potassium dichromate travel in millimeters? And how far did Janus green travel in millimeters also? And then we gave you a table for you to record it. So let's go over that table and maybe we can punch in the data together and then you can answer some questions concerning this part of the exercise. All right, so remember we said that we could compare these tubes, right? So here are the tubes at 5 degrees Celsius and here are the tubes at room temperature. And now look at, for example, potassium dichromate and how far it traveled at room temperature versus 5 degrees Celsius. And the same can be said for Janus green and even aniline blue, right? In each case, room temperature shows farther diffusion than 5 degrees Celsius. And yet, if you just focus at the tubes at 5 degrees Celsius, you'll see that aniline blue travels a short distance, potassium dichromate travels farther, and Janus green is somewhere in the middle. In every case, it's the same, right? Pretty cool. Okay, so let's put the data in there now. So find this table in your lab now, and I want you to record the 100 hours that we allowed the tubes to diffuse, okay? And then let's take note here of the distance that each dye traveled into the tube. Or I'm going to give you this data so that you can then process it and then answer some questions yourself. So how far did aniline blue travel into the jello when it was sitting at 5 degrees Celsius? Let's write 8 millimeters. Whereas potassium dichromate traveled 26 millimeters and Janus green traveled 18 millimeters. Okay, so all of this is at 5 degrees Celsius. Now, what about the set of tubes that were left at room temperature? How far did they travel in? Okay, well, aniline blue traveled 10 millimeters. And that makes sense, right? the temperature got warmer for aniline blue, so it traveled farther. Potassium dichromate traveled 29 millimeters, and Janus green traveled 21 millimeters. So what I'd like for you to do is, I want you to take the distance that each one of these traveled, and I want you to divide it by 100 hours. And what that's going to do is it's going to give us the diffusion rate in millimeters per hour. So for example, if you take 8 millimeters and you divide that by 100 hours, it's going to give you millimeters per hour. In this case, it's 0.08 millimeters per hour. And if you do the same for, say, potassium dichromate, at 5 degrees Celsius, it traveled 26 millimeters. In the course of 100 hours, that's going to be 0.26 millimeters per hour. Okay? And so that's what you're going to be placing here. I want you to do the math and fill in those cells in your table. Okay, once you've filled in those cells and you have the rate of diffusion, then I want you to graph it, right? So you're going to graph the distance traveled in those 100 hours. The distance in 100 hours, also for room temperature, right? So you'll do 5 degrees and you'll do room temperature. These are going to be bar graphs, okay? They're not line graphs. So you should have something like this. Let me clear up my y-axis here. You should have something like, okay, aniline blue, how far did that guy travel? And it might have traveled that far. Now you're gonna have to fill in your y-axis here, right, with the appropriate measurements. And then potassium dichromate, PD, maybe that one traveled farther. And then Janus green, you should have it somewhere like that, right? We want to have a visual representation, a graphical representation of what we see. And then for room temperature, right, let's try and keep the same sort of scale so that we can compare. And you'll see that aniline blue should travel a little bit farther than it did at 5 degrees. It should travel a little bit farther at room temperature. Potassium dichromate should have traveled a little bit farther. And then Janus green also would have traveled a little bit farther. So I want you to label everything, right? Make sure that you have a good scale here, a consistent scale, so that we can make a comparison between both of these bar graphs. Should be pretty simple. Again, email me if you have questions. Okay, let's look at the next exercise. This one covers osmosis. And remember, osmosis is the diffusion of water through a semi-permeable membrane until reaching equilibrium. This lab exercise is really cool. In this one, we use dialysis tubing. Dialysis tubing, like the name implies, is the sort of tubing that we would use in a dialysis machine. And some of you might know that dialysis is the process of filtering out nitrogenous waste from the blood for a patient whose kidneys are failing them. So we need to do it artificially, and we use dialysis machines to do that. The dialysis machines do the filtering with the use of dialysis tubing, and that's what we're showing here. So what you would have done is you would have taken a tube, and you would have sealed one end of the tube, left it open at the other end, and then poured in there a solution of like 10 milliliters of a specific concentration of sucrose. The specific concentrations are outlined in your manual. We're going to go over those here in this exercise as well. We use things like 15% sucrose and 30% sucrose. Okay. And then I'll diagram all of this in the next slide that follows. Okay, so once the bags have been assembled, then those bags are placed in a beaker, and the beaker would have either water or some other concentration of sucrose. And what we would want to see is in what direction water would flow. Okay, so I'm trying to give you visuals of what the lab setup looks like, but in the next slide, I'm going to diagram everything for you. You might want to look at table 5-3 of your lab manual and follow along with me. You can pause the video, get your lab manual in order, and then come back and follow along. All right. So let's look at the first bag, or sac number 1, as described in table 5-3. Okay, in this one, you would have a beaker filled with water, and you would put in here a dialysis bag, right? That would also have water. So the bag versus the beaker. The dialysis tube represents the semi-permeable membrane. Dialysis tubes are porous. They allow some substances through, but not everything. So in that regard, they act like our cell membranes. The bag in this case would have deionized water. So that's just basically 100% water. And you would place that in a beaker also with deionized water, which is again 100% water. All right, you should recognize these two solutions as being equal concentration. So this should be an isotonic setup, right? This is an isotonic set of solutions. They have equal water pressure on both sides, right? Okay. What about, this is okay, this is bag number 1. What about bag number 2? Okay, bag number 2 would also have a dialysis bag tied up at both ends, but this bag would have 10 milliliters of 15% sucrose. So if it's 15% sucrose, you should recognize that that means it's 85% water. The bag, in this case, would have deionized water, which is 100% water. So what do we expect in this case? Well, we would expect that the bag would start gaining water. How would we know that? We would know that because we would have weighed the bags at the very beginning before placing them in the beakers. And then every 15 minutes, we would pull them out of the beakers, and then we would weigh them again to see if they're gaining weight or losing weight. And what we would notice is that this bag right here is going to gain weight, whereas this bag right here should not, because it's in an isotonic solution. So, okay, let's practice now some vocabulary. How would we describe the bag that's gaining water? We would describe it as being hypertonic. And we would describe the solution that's losing the water to the bag as being hypotonic. All right, that's bag number 2. Let's look at bag 3. All right, same thing, a beaker with a dialysis bag tied up. But this time the bag has 30% sucrose. And, boy, I think I meant to say beaker here. Somebody should have said something. Okay, the beaker now has deionized water. Again, the dialysis bag represents the semi-permeable membrane. So 30% sucrose really means 70% water. And the beaker has 100% water. And water, again, would go from high concentration to low through the semi-permeable membrane. We would describe the bag as having gained water to be hypertonic. And the beaker that lost the water to the bag would be hypotonic. Right? Okay, here's a good question. Which bag, bag 3 or bag 2, would gain the water fastest? That's right, it would be bag 3. Why? Because bag 3 has a larger concentration difference, right? It's got a higher degree of concentration than bag number 2. So what's the difference here? The difference is only 15% difference between 100 and 85%, but the difference here is between 70 and 100. That's a 30% difference. So this is a higher concentration difference than this one, and that means that bag 3 should gain water faster, right? That's the degree of the concentration gradient as a factor that affects diffusion, in this case the diffusion of water. Okay, the fourth bag is a little different. The fourth bag has water, H2O. This is deionized water. And in this time, the beaker has the 30% sucrose. So that means that, the 100% water on this side is going to go towards the 70% water on this side. And now for the first time, the bag is hypotonic because it's losing its water to the beaker. The beaker is now for the first time hypertonic. All right, so this is what we would have noticed taking place in the course of 1 hour of monitoring each bag, right, every 15 minutes to see if it's gaining or losing water. All right, so let's look at our data tables and let's put some data in there so that you can crunch some of these numbers and answer some questions. Okay, so we've assembled the bags. We've placed, before placing them in the beaker, we decide that we're going to weigh them. This would represent their weight at 0 minutes. And here's the data. We would have found that bag 1 maybe weighs 11 grams, bag 2 weighs maybe 10.8 grams, bag 3 maybe weighs 11.2 grams at the beginning, and bag 4 maybe weighs 10.5 grams. You would have been weighing these with those digital scales that we have in the lab. But then after 15 minutes, we pull them out of their respective beakers, and then we check to see what their mass is. And maybe we find that it's 12 grams at 15 minutes for bag 1. It's 11.1 grams for bag 2, 11.9 grams for bag 3, 10.1 grams for bag 4. Now you might be wondering, hey, why did bag 1 gain water? It's in an isotonic solution. Well, remember that when you assemble the bags, you will have assembled the bags with dry string, but then once you place them in the beaker, the string gets wet, the string absorbs the water, and when you pull the bag out to weigh them after 15 minutes, you will have noticed now a slightly heavier bag because now it's got wet string. But maybe after that, we shouldn't expect too much of a change. All right, so we weigh them, we put them back into their respective beakers, and then we wait another 15 minutes, and now the total time elapsed is 30 minutes. We pull them out again, and we weigh them. And maybe we find this, 12.1 grams, 11.9 grams, 12.4 grams, 9.7 grams. And we repeat again at 45 minutes, 11.9 grams, 12.2 grams, 13 grams, 9 grams, and then finally, 60 minutes later, 12.1 grams, 12.5 grams, 13.4 grams, and 8.3 grams. All right, so take a glance at the data, see if it makes sense to you. Let's look at bag 1. Bag 1 looked like it maybe was gaining weight first 15 minutes, but then after that, it really didn't gain much or lose much. Kind of just seems like water is sloshing back and forth between the bag and the beaker. But if you look at the 15% sucrose bag, I didn't mean to cross it out. Let's go back. That bag seems to be gaining the entire time, and we expect that. The 30% sucrose bag, that one also seems to be gaining. But now look at bag number 4. Bag number 4 seems to be losing its water weight. All right, so what are you supposed to do with this data? Okay, we want you to record the percent change in mass over time. And we give you a formula in the next table. The percent weight change can be determined by taking your final reading. Let's go back to the original table so that we have the data. You're going to take your final reading and you're going to subtract from it your initial mass or reading, and then you're going to divide that by the initial. There's a couple of ways of doing this, and the way that I prefer to do it is to do it for every 15 minutes. So what I mean to say is that this column will always be your initial weight. And that means that at 15 minutes, this is your final, I'm calling it weight, but I should be calling it mass. This is your final mass. So let's just do this example here. What if we apply this formula, it would look like this. It would be final, which is 12 grams, minus initial, which is 11 grams, and then divided by your initial, which is 11 grams. So this would be basically 1 gram over 11 grams. And then that's going to give you your final minus initial times 100. You'll get a percentage that you can report in the next table right here. Okay, let's go back and see what we get for this one here. All right, in this case, you're going to take the mass at 30 minutes. That's your final. And what's always going to be your initial? The original mass at 0 minutes, right? So let's see now what happened when you compared 30 minutes to 0 minutes. It's 12.1 grams minus 11 grams divided by 11 grams, which is now 1.1 gram over 11 grams, and then times 100 to get the percent, and then that's going to go right here. And then repeat again for 45 minutes, subtracting that from the initial, and so on and so forth. And you're going to do that for each bag to fill in not just that row, but every single row. Okay? So you're going to have percent change in mass over the course of 1 hour in 15-minute intervals. And then what you'll do is you're going to graph it. So find this step or this figure 5-5 in your manual. You can add the missing 60-minute here if you want. And then you're going to have to use the appropriate scale here. Right? Fill that in. Why do we have a positive and a negative? This graph maybe looks a little strange to some of you. Well, I'm going to go back and I want to show you why. Do you see that bag number 4 is losing mass? Right? So this bag is probably going to show something like this. Right? Whereas bag number 1 is not going to show much change in mass. Bag number 2 is going to come up a little bit, and then bag number 3 should come up more than bag 1. So this should be bag 1, bag 2, bag 3, and bag 4 will be down there, right? So I just sort of loosely graph these, but yours are going to be more detailed because you're going to have the appropriate scale here and here to create your graphs. All right, again, email me if you have questions with this, and maybe I can send you some images or a brief video. I'm kind of getting good at this. Or maybe I'm not, I don't know. All right, let's go on to the third exercise. This one explores just how selective dialysis tubing is. So let's talk more about this tubing. What is it made out of? I want you to remember that your cell membrane is made out of phospholipids. And if you recall from our first part lecture of chapter 5, things that have a charge, they cannot get through the membrane. And things that are really large, they cannot get through the membrane. In fact, if you're small and you have a charge, you can't get through, right? You're going to bounce out. You cannot get through. But if you're small and you don't have a charge, you're going to get through just fine. Even slightly larger things, as long as they don't have a charge, something like cholesterol, that can get through too. All right, again, this is in the case of membranes. But what about the case of the dialysis tubing? Okay, the dialysis tubing is made out of cellulose. Remember that that's plant fiber. So we have technologies now that allow us to take fibers, cellulose fibers, and weave them together in such a way that these fibers can be interwoven so that there's these little pores now. And the size of those pores, or just how tight we weave the fibers, will determine what sort of things can get through. So we learned that water can get through. We learned this in the osmosis section of this lab exercise, right? But water is like, 3 atoms. It's not very big. What if you're bigger than that? What if you're like a polysaccharide like starch, something that was so massive we could see it under the microscope? Wow, so if water is 3 atoms, a polysaccharide is just huge. That'll never get through this bag, right? Okay, so that's basically what we're trying to show you here, the fact that membranes are selectively permeable, in part based on size. So the dialysis bag works for us as a decent model of what membranes are like. How does the dialysis bag fail in this model? The dialysis bag fails in this model because the dialysis bag would allow something very, very small like chlorine that has a charge. That will get through the dialysis bag, no problem. Would chlorine be able to get through a membrane? The answer is no. Chlorine is small, but it has a charge. That charge would be an issue for our membranes. But it's not an issue in the dialysis bag because the dialysis bag doesn't have a fatty layer to prevent the chlorine from getting through. That's the part of the dialysis tube that doesn't really work well. But as far as size, the dialysis tube is a good model for how membranes discriminate size. It's just not a good model for how it discriminates charge. All right, so let me go back. I'm getting too far along. So what's in the bag and what's in the beaker? All right, so the bag in this case would be filled with a solution that contains 25 milliliters of a solution that's mixed. The mixed solution contains 1% starch, and it contains 1% sodium sulfate. All right, so that's a mixture, right? We would just pour it right into the bag. The beaker would be filled with a solution that contains 1% albumin. Do you remember what albumin is? Brownie points if you do. Albumin is egg white, and you tested for it using Bio-Red in the macromolecule lab. It also contains 1% NaCl, sodium chloride. Okay, so remember I said starch is just a massive molecule. That's a polysaccharide. It's huge. That guy will never be able to get through the membrane. Albumin, that's also a macromolecule. That's why it was in the macromolecule lab. That's not going to be able to get through the membrane. So if we let this bag sit in the beaker for 1 hour, we're going to find that starch that was in the bag stayed in the bag the entire time and never ever ended up in the beaker. And the albumin that was in the beaker will never ever ever end up in the bag. It's just too big to get through. But what about the other stuff? Well, sodium sulfate dissolves in water to give you sodium ions, Na+, and SO4 with a 2- charge. Here, sodium chloride dissolves in water to give you sodium ions and chlorine ions. We're not going to look for sodium because sodium is on both sides of the membrane. We won't know who got through and who didn't, right? So forget that. Forget sodium. So what's left is sulfate, which is 5 atoms, and chlorine, which is 1 atom. And the question now is, can chlorine get through? And the answer is, what did we say? Yeah, no problem. Because chlorine is only 1 atom. Right? Remember that chlorine, even though it's 1 atom, our membranes will discriminate because chlorine has a charge. But in this case, here in the dialysis tube, the tube does not discriminate charge. So what we would notice is that the beaker had chlorine, but now we're going to find that chlorine has entered the bag, gets through, no problem. And sulfate, which was in the bag, SO4, 2 negative charges, it's 5 atoms. What do you think? Can that one get through? Water got through. Water was 3 atoms, if you remember. What about sulfate with 5 atoms? Can that get through? Well, it can, but do you think it can get through as easily as chlorine, which is 1 atom, right? If the pores are that big, maybe chlorine zips right through, no problem. But sulfate's going to have a little bit of a hard time, but it'll still get through. It just won't be able to get through as easily as chlorine. All right, so how do we test to see if they got through or not? Well, we have some biochemical tests to perform, and you can read about those tests in your manual. So 1 hour after setting up the beaker with its bag, you would then pull the bag out of the beaker. You would take samples of the solution in the bag, and you would test the bag solution for things like starch, albumin, sulfate, and chlorine. And then you would take the samples of the beaker fluid, and you would test it also for starch, albumin, sulfate, and chlorine. So let's look at that data table. Find these data tables in your manual. And then let's look at what was in the beaker. Right? So at the beginning of the experiment, was there starch in the beaker? No, there wasn't. You can review this film and go back and see what it looked like. After 60 minutes, we would find that no, it didn't get through. Do you remember how we test for starch? Brownie points? That's right, we use iodine. Everybody answered correctly. This is what's amazing about video. Okay, what about sulfate? Sulfate was not in the beaker, right? Here's that negative sign saying no, it wasn't in the beaker. But after 60 minutes, we would find evidence that it got through. We would see a precipitate forming. What about the chloride ion? It was in the bag, in the beaker at the beginning, so we should find that it's still in there, right? It's not like all of it left. It reaches equilibrium. That doesn't mean all of it leaves. It's going to be in both places in equal amounts. Albumin was in there, so it should be in there. So let's look at our data here, our result data. Everything that was present in the beaker is still present in the beaker. But now we find that sulfate wasn't there, but now it is. So sulfate was able to get through. And what we've learned about the dialysis bag is that it allows sulfate ions to get through because the pores of the dialysis tube are big enough to let the sulfate ion through. how would we have tested for sulfate ion? We would have used barium chloride. Again, you can read about this in your manual. I'm trying to keep at minimum all my talking. Chloride ion, you would use silver nitrate to test for the chloride ion. And then do you remember how we tested for albumin, a protein? We would use biuret. So you should recognize biuret and iodine because you have experience with those. But these two, these are brand new. Okay, let's now look at the dialysis tubing, the bag. Starch was in it at the beginning, so starch should be in it at the end. Sulfate ion was in there at the beginning, so it should be at the end. Chloride ion, it wasn't there, but we agree that it was small enough to get through. And then albumin, it wasn't there at the beginning, still not there at the end. So I want you to go back to your tables and I want you to insert this data, but I need you to be able to tell me how you tested for these to determine if they were able to get through the bag or not. All right, we're getting now to the fun part of the lab exercise set. In this case now we apply what we've learned about diffusion and osmosis to actual living cells. And we would have used Elodea, the aquatic plant that we have growing in the lab. We would have used Elodea to see how it responds to different solutions. So in this case, we would have given you two slides, slide A, slide B, and you would have taken a small leaf of Elodea. Oh, maybe I should make that green, right? All right, so here's my Elodea, and I'm going to put one leaf on each slide. And then on this one, I'm going to add a drop of deionized water, which is, again, I'll remind you, 100% water. And on this one, we would have added 20% sodium chloride. All right, so this one is in a salty solution, and this one is in a 100% water solution. And then we would have put a cover slip on top. And then we would have said, okay, have fun, look at it under the microscope. What would we have seen? Well, if you look at the cells of Elodea in 100% water, the cells would have all of these chloroplasts swimming right around the edges. These are super happy plant cells, right? And that's because their central vacuole is filled with water, right? So how do we know that? Well, the cell can now be compared to the deionized water. This is 100% water. The cell has a bunch of dissolved substances inside, ions, sugars, all kinds of things are dissolved in there. So it has less water than 100%. So water would have gone into the cell, it would have bloated the central vacuole, and because the central vacuole is in the center, it would have filled up and pushed all of the chloroplasts right onto the edges, and that's why you would have seen all of these chloroplasts swimming around just beneath the cell wall. I'll show you images after this slide. Okay, but what about the Elodea that was placed in a salty solution? Okay, so that one's cell wall would still be nice and big, but then we would have noticed that the cell membrane has begun to peel away from under the cell wall. The central vacuole would appear to be shriveled, and all of the chloroplast would be dragged to the center. And we would also notice all of these air spaces right here. So you should recognize this from one of the videos in the first presentation for chapter 5. Okay, so this plant cell is being plasmolyzed, which means it's being dehydrated. This is going to be a wilted plant. On the other hand, the cell up at the top with deionized water, this one's bloated. And we describe bloated plant cells as being turgid. Turgid means bloated. Remember, our animal cells, we don't like to be bloated. For us, that would be like hypertension sort of stuff. But plant cells can afford to be bloated because they have something we don't. You remember what that is? That's right, cell wall. Cell wall is amazing. We don't have it, so we can't afford to get bloated. Plant cells, they're cool with it. All right, so let's look at some images of what this would have looked like under the microscope. Yeah, look at that. Okay, so here's the cell membrane completely peeled away from the cell wall of the plant cell. And look at all this airspace, right? And all the chloroplasts are somewhere in here now, dragged to the center. Whereas in a happy plant cell that's in 100% water, the central vacuole is somewhere in the center, right? You can't really see it, somewhere in here, right? But it's filled with water and it pushes all the chloroplasts to the edges just underneath the cell wall. And they're swimming around, they're having a good time. All right, so that's the difference between salty water and 100% water in the case of plants. What about our animal cells, like red blood cells? Okay, so for us, osmotic changes in red blood cells, we would have asked you to create 3 slides. And you would have added blood to each slide. And then each one of those slides would have received a different solution. This one would have had deionized water, maybe. This one here would have had maybe 0.9% sodium chloride. Does anybody here work in the medical field? You should recognize 0.9% sodium chloride. This is basically what you find the concentration of salts to be in IV bags. All right, think about why that's important, right? IV bags have a fluid that you are delivering straight into the bloodstream of a patient. What if you delivered the wrong concentration of fluids? What's going to happen to their red blood cells? Okay, keep thinking about that. The third slide would have 10% sodium chloride. And then you would have put your cover slip on top. And you would have looked at them under the microscope. So what do cells look like when you place them in 100% water, such as deionized water? Well, those are going to feel and look bloated. And they're kind of difficult to see, in fact, because once they start filling up with water, the hemoglobin that gives them their red color starts leaking out. So they start basically looking like just cell membranes filled with water, and for that reason we call them ghost cells, because they're really hard to see. But I'll show you images of what they would look like with a better microscope. Okay, what about slide B that has 0.9% sodium chloride, something that's a little bit of salt, but not a whole lot? Those red blood cells would look happy. They would be thick on the outside, thin in the middle. They're doing great. All right, now do you understand why IV bags have 0.9% sodium chloride? It's because that's the exact osmolarity of our bloodstream, right? It's actually 0.85 or something like that. So what this means is that water is leaving and water is entering at the same rate. And so these cells don't experience any change in water pressure. They don't bloat, they don't shrivel. Okay, but what if you place a red blood cell in a solution that is 10% salt? Well, in that case, it's going to start to shrivel up. And you'll see all kinds of shriveled up cells. And they have these little pointy things. Those are basically proteins that are congregating they're sticking to each other on the cell membrane. All right, so how do we describe all of these cells? We have terms for these cells in your manual. We give you a diagram, right? And we show you images of each, and we describe what they look like. So this one here would be hemolyzed, right? Destroyed. These would appear normal, and these would appear crenate. All right, let's go over our tonicity terms. If the cell is bloated, like in this case, then the cell must be hypertonic, right? And that means that the solution must be hypotonic. All right, so I want you to remember that 100% water is always hypotonic. Okay, what about in the case of 0.9% sodium chloride where water is going in and out at the same rate? We would say that in this case the solutions are isotonic. And what would we say if the cells are losing water? Then the cells are hypotonic. And the solution that was gaining the water is hypertonic. All right. There you have it. So all you have to do now is complete the questions in your manuals, including the post-lab questions, and wait for further instruction on how to submit those so that I can grade them for you and give you some scores. All right, y'all. Have a good one. Oh, wait, no. Here's the images. Here's the images of those cells. I almost forgot. I thought my diagrams would be enough. They're not. Okay, here are the cells that are in deionized water. Notice that you really can't see the thickness on the outside and the thinness in the middle. They don't have that donut shape to them like these do. These are the normal cells. And then over here are the crenate cells. These are the ones that have shriveled up. All right, almost forgot about these. And